Hi everyone, my name is Natalie and I'm currently a postdoc at UCLA, but today I'm going to talk a bit about work from my PhD, which used imaging spectroscopy data from shift to investigate grazing effects on vegetation. To provide some background on shift, it stands for the Surface Biology and Geology High Frequency Time Series. The goal of it was to prototype data and algorithm calibration for the upcoming planned NASA hyperspectral satellite, SBG. And to do so, hyperspectral images were collected about every week for a period of about 3 months. And at the same time, we collected in situ data on or around each flight date. So on the right, you can see a map of generally the flight box in red and our main terrestrial field sampling sites, which were focused at the Dangerman Preserve, the Sedgwick Reserve, and a little bit in Los Padres National Forest. And during that time, we sampled over 100 unique species. You can see the full phylogeny on the right of the species we sampled. And again, I have that same map on the left, but now showing the distribution of our grassland versus shrub versus tree plots. And we had over 480 unique plot locations, with some of these being resampled through time. Now, at each plot, we would characterize the dominant vegetation species and collect foliar samples of those species. And we would send the foliar samples off for chemical analysis to retrieve some certain foliar trait data. And at the same time, we got GPS points, of course, of the plot locations and extracted the spectral reflectance at each plot. And the benefit of having now these trait data and the spectral reflectance is that within the image, there are certain absorption and reflectance features that relate to these certain foliar traits. So with all that information, we can build models and then map out the foliar traits across the landscape. And this is really the first data set of its kind where we have this dense hyperspectral time series and create these maps throughout the season. So this is looking at the distribution of foliar nitrogen across the Dangerman Preserve and Sedgwick through the period of the SHIFT project. And with multiple traits, we can also start to look at how the landscape differs along different trait axes. So for example, I have 3 traits shown here: leaf mass per area in green, nitrogen in blue, and phenolics or defense compounds in red. And it really highlights the different functional strategies across the landscape. Now, I'll be leveraging these trait maps, and my focus in particular is to look at how grazing affects the foliar traits. And Sedgwick and Dangerman both provided good, unique opportunities to do so. At Sedgwick in particular, we have a really long history grazing record, and that helps us look at legacy effects of the effects of grazing. Whereas at Dangerman, we have really detailed information on the timing and the intensity of the grazing, which can help assess some short-term effects. So at Sedgwick in particular, I separated the landscape into 3 broad groups. We have up in the coral, the area that was grazed for about, or ungrazed for about 20 years and then grazed for the year preceding shift. And then in blue, an area that was ungrazed for about 20 years. And then in green, an area that was grazed for about 20 years, but probably much longer than that. And for simplicity, I call those the new graze, the ungrazed, and the old graze group. So again, remember, new graze just means it was grazed for a short period of time, and old means a long period of time. And in contrast, at Dangerman, for the year of shift, we have information on the timing and the amount of cows moving in and out of each pasture. So here I'm showing the animal unit months used in 2022, which I'll use as the indicator of grazing intensity here. Where the darker green indicates the higher intensity grazing. And my test will focus on these 2 pastures in particular, which are neighboring each other. The one on the left had higher intensity grazing and was also actively grazed for a few months leading into the SHIFT project, whereas the one on the right was lower intensity grazing, although it also had active grazing during SHIFT starting near the end of the period. So these 2 I'll call heavy versus light grazing. So again, the focus at Sedgwick is more the length of time in years versus at Dangerman, where the focus is more the intensity of the year of shift. And to do these comparisons, I first mapped out areas that weren't grass. I'm only focusing on grassland areas in this test. And then I stratified the landscape based on topography and soil type. As I saw just in the literature, these are main drivers in grassland foliar trait distributions in California, at least. And I conducted 2 different tests to compare the different pastures. One is spatially correlated functional ANOVA. That's really for comparing groups of time series. And the other is principal components analysis, which is a dimensionality reduction technique. I'm going to briefly explain how this test is conducted because it's probably new to most of you. The spatially correlated functional ANOVA, especially, this is just example data. We have, or essentially, we have 2 different groups and we have pixel-wise time series for each group. So those are shown here. And in the first step, I smooth those pixel-wise time series so they're all represented now as functions, and we can directly compare these different functions. And I obtain a mean curve for each group. Then I take the difference between those mean curves, that's shown in black here, and I compare it to a resampled interval of what those differences might look like under the null hypothesis. After this, of the smooth curves and the difference. And what you want to look for, the smooth curve will help us see how the phenology is characterized through time of that trait, and the difference will help us understand like where the black curve is outside of that resampled interval. That's where the groups are significantly different. So if we focus on Sedgwick first, I've pulled a couple traits as an example. On the left, we can see the phenological trends of foliar nitrogen and leaf mass per area, where there's this clear trade-off between LMA and nitrogen. Nitrogen decreases throughout the season, LMA increases. But if we look at the direct comparison of the old grazed or so long-term grazing group to the ungrazed group, we see that a longer history of grazing is leading to lower foliar nitrogen and higher leaf mass per area. And if we look at a couple other traits as well, we can see that longer history of grazing is leading to lower phosphorus and lower potassium. But the value of this visualization is it helps us see that that pattern is not necessarily true the whole season. So phosphorus, for example, becomes more different later in the season. And then if we take a look at the principal component analysis results, essentially the different components are summary indices that help us see how our observations are distributed across the multi-trait space. So now we don't have to just compare one trait at a time, we can see the whole picture. And we see all of the observations are moving from this higher nutrient concentration to higher structural investment through the season, but there's still this distinct grouping of our different grazing treatments where longer history of grazing is weighing more heavily on structural investment like leaf mass per area and leaf cellulose. Now, if we pivot to Dangermond results, we'll see that same trade-off we saw before in nitrogen LMA, for example, where nitrogen decreases, LMA increases. However, the difference between the groups is a bit less pronounced and also in a way the opposite of what we saw before. So I've marked in vertical lines here the period that the point where the heavy grazing period ended in the first pasture and the light grazing period began in the other. So you can see, for example, the heavier grazing seems to lead to higher nitrogen and lower leaf mass per area, but not until the grazing period stops. And a bit of a different picture here too for phosphorus and potassium, where leaf phosphorus is higher where there was heavy grazing. And for potassium, the difference is not so pronounced. And now I'll show once again principal component analysis results for the Dangermond pastures, where we can see that same pattern of, in general, all observations moving from high nutrient to high structure. But what's interesting here is we don't see that same separation for the whole season. Instead, we see the two groups are more different early in the season and in the middle and become very similar near the end. So ultimately what I found is that the grazing effects really depended on the context of the grazing itself. So we see these really pronounced differences at Sedgwick, where again, we have a very long-term consistent history for these different pastures, versus at Dangermond, where there were still differences. They were maybe less pronounced and they were maybe more short-term. So these grazing cycles could drive short-term increases in nutrients or causing that lower leaf mass per area like we saw. there's a lot more to the story than what I can show here, of course. There are other environmental gradients to consider beyond just soil and topography, for example. There's also species differences that itself could drive differences in functional trait composition. And of course, there's species differences driven by grazing itself. And finally, there's this clear accumulation of senescent plant material like old dead grass that builds up with a lack of grazing. We've seen that at Sedgwick for sure, at least, and we have to investigate more how it might affect our trait estimates. This work built upon the effort of a lot of people, which I'd like to thank. I also wanted to mention that the overview paper for Shift was just published in Ecosphere, so I definitely recommend checking that out. And I'd like to thank everyone at my old lab in Wisconsin and my new lab at UCLA for their support, and the staff at Dangermond and Sedgwick for helping me a lot understand the different grazing histories, and the whole Shift team, of course. And if I have time for questions, I'm happy to take any. Okay, thank you.
Really cool. Digestion, something I take away from your results is there's kind of a juicy to crunchy continuum.
Yes.
Of plant features. I guess I wonder, have you had a chance to talk with grazing operators and land managers on the ground about what this means potentially, or how this insight can be used for understanding long-term seasonal management?
That's a good question. Not really, beyond talking to Frank at Sedgwick and Moses at Dangermond. Not too much about the actual grazing operations themselves and how it could inform, let's say, like the timing or like the locations for grazing. That's something I would love to do, though. I think there's great potential for like integrating it into management, at least brainstorming ideas for management. Yeah. Hi, yes.
Thank you so much, Natalie. That was wonderful. Do you have any insight into the climate differences and how that might affect the nitrogen availability over those sites?
Do you mean the climate differences between? Yeah, no, that's such a good idea, or such a good question. I have considered it only a little. I originally, this was right at the end of my PhD, so it originally was just analysis for Sedgwick, and I was able to incorporate Dangermond later on. For Dangermond in particular, I had difficulty trying to decide which pastures to focus on because I felt like that proximity to the ocean, I'm not so familiar with how that's going to be affecting things. So I picked these two right next to each other. I have a feeling that there's going to be a big difference in yeah, how the climate is affecting the results there, but I'm not really sure what it could be. So for example, I see where there's higher intensity grazing, there's higher nitrogen, let's say, at Dangermond. Maybe it could just be that there's better conditions because of more moisture in the air or like more temperate climate. It could be good conditions for like a grazing to then cause this burst of growth later. Whereas maybe at Sedgwick, if it's drier later in the season, I'm not sure, maybe we wouldn't see that. Yeah.
Well, and just for everybody, is that soil moisture matters in nitrogen cycling. And so you're going to get a different moisture regime on the coast than you'll have here. So I think that you've started something that I think others should hang on to.
Yeah. Yeah. Thank you.
Thank you, Natalie.
Yeah. Thank you.